Hello students, today we will discuss about the C4 pathway in photosynthesis and the related anatomical features, that is Kran's anatomy. Clear? So C4 pathway and Kran's anatomy is the topic for our today's discussion. Now we know that in tropical region the conditions like high light intensity, clear high temperature, high carbon dioxide concentration, all these are the conditions which are prevailing in the tropical areas, and these are very favorable for photorespiration. And we know that photorespiration is absolutely a destructive process. Clear, it may C4 in tropical region, these conditions favors photorespiration so much that if photorespiration occurs in normal way, then most of the carbon which is fixed during biosynthetic pathway will be released by photorespiration. So to avoid this destruction, clear, which may lead to even the death of the plant. So to avoid this destruction, these tropical plants like maize, sugarcane, millet, all these plants, clear, they have adapted a special pathway which is called C4 pathway. And as this pathway needs certain changes, so they have some anatomical features, clear, which is comparatively different from that of the C3 plants, and that particular anatomical features which these C4 plants have, they are called the Kran's anatomy. So these anatomical feature which the C4 plants have, clear, these are called Kran's anatomy. So first of all, let us see what anatomical differences or what anatomical special features do the C4 plants have. Now, what type of anatomical features we will see in the C4 plants? Number one is the mesophyll cells. If we go on for the mesophyll cells, we will find in C4 plants, mesophyll cells is not differentiated into palisade and spongy parenchyma like that of the C3. So, undifferentiated mesophyll cell. Clear, this is one of the feature. Next, the chloroplastid which are present in the mesophyll cells, they are comparatively small. Clear, they are comparatively small and contains grana. Clear, that granastroma part is very less. So first of all, the mesophyll cells, they are undifferentiated mesophyll cells in C4 plants, and the chloroplastid which are present in these mesophyll cells, they are comparatively smaller, and grana here is very well developed. Whereas in C3 plants, we are supposed to see that mesophyll cells is differentiated into palisade and spongy parenchyma, and these chloroplastids with grana and stroma are well developed in the mesophyll cells. But here the grana is more developed. Clear, chloroplastids are comparatively small and grana are more developed. Next, if we go for the bundle sheath cell, we will find that in C4 plants, bundle sheath cells of the C4 plants are comparatively larger and are of several layers, but where in C3 plants they are comparatively smaller and one or two layers. But in C4 plants, bundle sheath cells are comparatively larger cells and have several layers. Next is the chloroplast. If you see the chloroplast here, they are also larger. They have larger chloroplast, and that is without grana, because these bundle sheath cells, they are mainly for the biosynthetic pathway. These chloroplastids which are present in the bundle sheath cells in C4 plants are mainly for the biosynthetic pathway. So they do not require grana or that grana containing chlorophyll for fixation of light. Clear? So they do not require that. They are mainly meant for biosynthetic pathway, and the photochemical portion is done by the mesophyll cell. So in C4 pathway, in the plants with having C4 pathway, the photochemical, these mesophyll cells are supposed to do the photochemical path and the fixation of CO2. And bundle sheath cells with larger chloroplastid without grana, they are mainly supposed for the biosynthetic phase. That means conversion of the CO2 to carbohydrate. In this mesophyll cell, for fixation, the chloroplastid, the stroma contains phosphoenol pyruvate and phosphoenol pyruvate carboxylase for the fixation of CO2. Clear? And Rubisco is absent in the mesophyll cells. Whereas Rubisco and RUBP, that is Rubisco ribulose bisphosphate carboxylase oxygen, is that enzyme, and the RUBP ribulose bisphosphate is present in the grana, in stroma in the bundle sheath cells. And here, when this carbon dioxide is taken into the bundle sheath cells, which are fixed by mesophyll cells, are finally taken into bundle sheath cell, there the carbon dioxide is released, and finally this Rubisco and RUBP is used to form the carbohydrate. So in C3 plants, so how will we differentiate? In C3 plants, we will see palisade and spongy parenchyma. In C4 plants, the mesophyll cells are undifferentiated. There is no such differentiation of palisade and spongy parenchyma. In C3 plants, the mesophyll chloroplastids are larger with well-developed grana and stroma. But in C4 plants, the mesophyll chloroplastids are smaller with well-developed grana. Clear? In C3 plant, the bundle sheath cells are smaller with single one or two layered, but in C4 plants, bundle sheath cells are larger with numerous layers. In C3 plants, the chloroplastid of the bundle sheath cells are not differentiated, they are with grana and stroma, but in C4 plants, the chloroplastid of these bundle sheath cells are larger without grana, that is, agranal chloroplastid is present in the C4 plants. So this different anatomical feature which is present in the C4 plant is called Kranz anatomy. So what is Kranz anatomy? Is this feature which we find, that is mesophyll cell with smaller chloroplastid, undifferentiated mesophyll cell with smaller chloroplastid and well-developed grana, numerous bundle sheath cells, larger cells with larger chloroplastid without grana. So all these features which you find in the C4 plants is called Kranz anatomy. So let us see what happens in C4 pathway. So think this is the mesophyll tissue, clear, where we find chloroplastids which are smaller in size and they are well-developed grana, and this is the bundle sheath cell with which are larger cells with larger chloroplast but are granal, that means grana is not there, instead the whole stroma is there. So in mesophyll tissue, the chloroplastid of this mesophyll tissue does not contain Rubisco, the enzyme, clear, and RUBP to fix carbon dioxide. Instead, what they contains, you can easily see phosphoenol pyruvate is the substance, clear, which is used for fixation of CO2, and the enzyme is phosphoenol pyruvate carboxylase or PETcase. So phosphoenol pyruvate is the substance, clear, where the CO2 is who fixes CO2, and the enzyme regulating this fixation is the phosphoenol pyruvate carboxylase. Now phosphoenol pyruvate itself is a 3-carbon compound. When it combines with carbon dioxide, it forms an oxaloacetate, which is a 4-carbon compound. This oxaloacetate, this NAD, hydrogen is released from it by the reducing agent NADP, and finally it forms the malate. Malate, through this plasmodesmatal connection, moves to the bundle sheath cells. In the bundle sheath cells, this malate releases carbon dioxide and it forms the 3-carbon compound pyruvate, and therefore it forms phosphoenol pyruvate. Clear? And ATP is utilized during formation of this phosphoenol pyruvate, and because with the release of ADP. And this carbon dioxide which is released from malate is utilized by ribulose bisphosphate and rubisco for fixation, and ultimately the Calvin cycle occurs. Clear? So thus carbohydrate is generated from this carbon dioxide. So in C4 pathway you can see that mesophyll tissue has the chloroplast which is only to fix carbon dioxide, but the formation of carbohydrate from carbon dioxide, this occurs in the bundle sheath cells where there is rubisco, RUBP are present, ribulose bisphosphate and ribulose bisphosphate carboxylase oxygenase, the enzyme is present. Clear? Where the carbon dioxide is refixed, and finally it undergoes Calvin cycle to generate carbohydrate. So C4 pathway, since rubisco is not there in the mesophyll tissue, as a result what happens, the rubisco cannot be affected by the environmental condition, because we know when there is more carbon dioxide in the environment, when there is more light intensity, when there is more temperature in the environment, then the rubisco acts as oxygenase rather than carboxylase. So since it is very deep inside, clear, which is not affected by the environmental condition, rubisco here act always as carboxylase, never oxygenase, as because first of all it cannot be affected. Clear? Since it is very deep inside, it is not directly affected by the environment, and constantly as malate is releasing carbon dioxide in this bundle sheath chloroplastid, so within here the carbon dioxide concentration is absolutely always, it is high. So rubisco here never acts as oxygenase, rather acts only as carboxylase, and thus is more efficiently it forms carbohydrate. Clear? In C4 pathway, more efficiently carbon dioxide is utilized for formation of carbohydrate. But the question is that if it is so efficient, still why not the most of the plants are adapting this C4 pathway? Instead they are still adapting the C3 pathway. Clear? As because C4 pathway require more amount of energy for fixation of for formation of one molecule of glucose. C4 pathway needs 30 ATP, which is much higher than what is C3 pathway requires. C3 cycle or Calvin cycle require only 18 ATP. So the cost in relation to energy is much higher in C4 pathway, which most of the plants cannot afford. So that is why C4 pathway is not very common in all plants. Clear? The plants, since it is C3 pathway is more cost effective, so most of the plants follow C3 pathway instead C4.